Hello guys, I am Shubham from Show Tutorials. This video is about eukaryotic transcription PYQs. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. This is absolutely free from your side, but this will help us grow. So all about eukaryotic transcription we have studied. You will find the videos in the playlist Molecular Biology. So before we start with the PYQs, I will show you the two important charts which are very important, right? Please save this taking the screenshot. Okay, ये बहुत important है. First one is this one and second this. So you will see lot of repetitions from this pictures, right? Three types of RNA polymerase: RNA pol 1, RNA pol 2 and RNA pol 3. RNA pol 1, all the rRNA genes, okay, rRNA genes. Only the 5S rRNA is in the RNA pol 3, right, and tRNA. RNA pol 2, all protein coding genes, SNO RNA, miRNA genes, siRNA genes, long non-coding RNA genes, and most snRNA. RNA pol 3, some snRNA gene and genes for other small RNAs. Right? RNA pol 1, 5.8S, exactly 18S, and 28S rRNA genes. This picture is much detailed. RNA polymerase 1, free rRNA, okay, free RNA, rRNA, 28S, 18S, 5.8S rRNA. RNA pol 2, mRNA, snRNA, this is splicing, right? mRNA encodes protein. siRNA, chromatin mediated repression translation control. miRNA also translation control. RNA polymerase 3, tRNA, 5S rRNA, snRNA U6, right? U6, 7S RNA, other small stable RNAs. Okay, you can see the functions: protein synthesis, ribosome component protein synthesis, RNA splicing, right? Then signal transmission particle for insertion of polypeptide into endoplasmic reticulum, and stable RNA, various function unknown for many. Okay, now we will start with the questions. Very important. First question: alpha amanitin is a fungal toxin. Okay, fungal toxin है which inhibits eukaryotic RNA polymerases. The three eukaryotic RNA polymerases show differential sensitivity to this toxin. Means the sensitivity differs. Which one of the following order higher to lowest is correct in respect of sensitivity towards alpha amanitin? So RNA polymerase 2 is a direct target. Okay. So RNA polymerase 2 should be first. Okay. But after RNA polymerase 2, RNA polymerase 3 should be the next one. And then RNA polymerase. And so this is a direct question, you have to remember this concept. Right? Alpha amanitin sensitivity to eukaryotic RNA polymerases. RNA pol 2 is the first target, then 3 and then 1. Next question. In eukaryotes, precursors of microRNA, miRNA, see, and small interfering RNA, siRNA, are usually synthesized by. Okay, from that chart, अभी हमने देखा RNA pol 1 and pol 3 no, 3 and 1 no, RNA pol 1 no, RNA pol 2 yes. Okay, RNA polymerase 2, mRNA, miRNA, siRNA. A highly specific inhibitor that targets the phosphorylation activity of TF2H. TF2H is a transcription factor, initiation factor, right? Is added to an in vitro transcription reaction. In vitro transcription reaction में add किया है, inhibitor है of phosphorylation activity मतलब TF2H phosphorylation activity target किया है inhibitor which will stop the phosphorylation activity. Which one of the following step is most likely to be affected? So TF2H phosphorylation activity is related to promoter clearance. Okay, option 2, promoter clearance. And TF2H also has helicase activity which will cause promoter melting. Right, and promoter melting से क्या होगा? Close complex to open complex. Close complex to open complex formation. Right, but this question is asking about phosphorylation activity of TF2H, which is basically related to promoter clearance. Next question, which one of the following statements is generally true about RNA polymerase 2? Again RNA polymerase 2 का पूछा है. You can see the use repetition. It is dedicated to transcribing RNA from single transcription unit, generally a large transcript, which is in process to yield 3 types of ribosomal RNA. No. Okay, RNA polymerase 1 is responsible for ribosomal RNA. Second, it transcribes varieties of small non-coding RNAs which are expressed in all cell types. No, non-coding RNAs नहीं करता transcription. Third, it generally synthesizes various types of mRNA and small non-coding RNAs. Yes. So option 3 is the what is exclusively involved in synthesis of RNA and tRNA. No. This is RNA called 3. Okay, so option 3 is the answer. Next question, which of the following are not transcribed by RNA polymerase 2? Okay, RNA polymerase 2 ट्रांसक्रिप्शन नहीं करता. mRNA and snRNA करता है ट्रांसक्रिप्शन. mRNA, snRNA, yes, transcribes. mRNA and snRNA, yes. tRNA and 5S rRNA, this will be the answer. You have to see the not transcript. Okay, so tRNA and 5S RNA are transcribed by RNA polymerase 1, not by RNA polymerase 2. Given below are 4 sentences with blanks labeled X, Y, Z and L. Okay. RNA polymer transcribed X, mRNA genes are transcribed by Y. The RNA polymer is found only in plants with Z. rcRNAs are synthesized by L. Okay. Now, which one of the following options would present the combination of all terms in order X, Y, Z and L to complete the above sentence correctly? Right. How to solve this question easily in the quick manner? Let's see. miRNA genes to be है transcribed by Y. The Y should be RNA polymerase 2. So RNA polymerase 2 present in option 1 and option 3. Okay. 2 and 4 eliminate. Right. RNA pol 1 transcribe X. X should be mRNA. No. 45S rRNA. So option 3 answer होगा. Okay, like this you can solve very easily. RNA polymer is found in plants is 4 and 5 plants में होता है, right? And CSIR are synthesized by RNA polymerase. Option 3 is the answer. This is how you can solve quickly within few seconds. tRNA genes of a gram-positive bacteria were sequenced. Okay, tRNA genes, while some of the genes possess sequence corresponding to CCA end of the tRNA. We know CCA end present होता है, right? That is the tRNA, just a love diagram. This is a 3' end, right? 3' end which contains septal stem CCA. So some have this sequence, others did not. Interestingly, even the genes that lack the sequence means इसमें sequence present नहीं है corresponding to the CCA end of the tRNA were found to code for functional tRNA. Okay, means functional tRNA code 4 है. Why? This is because the CCA end in the tRNA is not essential for its function. No, it is essential. Okay. On the 3' end, this A यहाँ पे amino acid bind होता है, right? During the process of translation. So this is important. The genes that do not possess sequence corresponding to the CCA ends of the tRNA are required prior to the transcription. So we have not read as such. Okay, ये हमने पढ़ा नहीं है. The CCA end is added to the tRNA during their maturation. Okay, so this option is correct. CCA end maturation time add होता है. tRNA maturation or processing we can say. The primary transcripts of the genes lacking sequence corresponding to the CCA end are subjected to guide RNA mediated editing prior to their maturation. Okay, option 3 correct. Next question, same type of question repetition है. You can see in bacteria many of the tRNA genes do not contain the CCA sequence. CCA sequence present नहीं होता in some tRNA found at the 3' end of tRNA. In this context, which one of the following statement represent correct explanation? In these tRNAs, amino acylation occurs at 3' end of the tRNA irrespective of the presence of CCA sequence. No. amino acylation के लिए important है CCA sequence. Okay, CCA sequence is added to this tRNA transcript in a DNA template independent manner. Yes. Option 2 is correct. DNA template independent manner से add होता है CCA sequence. Let's check other options also. This tRNA exploit the process of transplacing to include a CCA sequence at the 3' end. Transplacing no. Okay, maturation से होता है. DNA template independent manner is the answer. The absence of CCA sequence occurred only in the large common ancestor during the course of evolution and current day tRNA gene always is extreme. Okay, ये confirm गलत होगा. Mostly possess a sequence to encode the CCA end of the tRNA. No, always possess is the extreme state. Right? This is why option 2 is correct. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel and stay tuned for the upcoming videos. Thank you.